So before discussing the trials themselves, we've talked about this a lot today on the panel and other things. I just wanted to give an example of two different patients with neuroendocrine cancers. And I do this because it's really tricky. I absolutely love when we have patient advocacy groups like this come together. But one of the problems is our disease is quite heterogeneous. And so not all of our patients are the same, right? So we always want to be able to make sure we're not missing anything. But sometimes the therapy is completely different because of the biology and because of the type of cancer. So this first patient is a 58-year-old gentleman who had very nonspecific abdominal pain for a day. He goes to the emergency room because it was pretty significant, but in retrospect really had no other associated symptoms. And he gets diagnosed with a well-differentiated neuroendocrine tumor, low grade. And as you can see here on the sort of cross-sectional imaging, those little white dots in his liver are the neuroendocrine cells and the tumors. And so you could see that they're kind of scattered there on both sides of his liver, and they're pretty small. So, you know, there are many different approaches here, and sometimes in an unsettling way. We always say definitely get a second opinion, but you could get 5 different opinions from this patient. And so that's a little, again, it's unsettling sometimes because, you know, and there's not to say there's a right or a wrong answer per se, but with this patient, we actually decided to just watch him for a little bit because the test of time in a low-grade tumor with low-volume disease like that can often help us a lot in better understanding the tumor itself. So anyway, so this patient has actually been my patient, and I just watch him every 6 months, and we're going 36 months without disease progression. So an example of very indolent disease, no somatostatin analogs, no symptoms, didn't need me, you know, and so I'm sort of putting myself in front of this patient to say, don't do anything until we have to. So it's not if we treat, it's when we treat almost always. But this biology allowed me not to do much. In contrast, case 2, I'm going to show you is a patient that was not the case. Actually did have symptoms that did not go away after one day and had a pancreatic neuroendocrine cancer and biopsy proven. He had a pancreas mass and bilobar liver disease, and I'll show you the next slide is the picture of his tumors. So that's his liver. All that white disease is cancer. Very different pattern, very different biology. Those white things next to that is something called varices, so he probably had this for a long time. But this patient got sick, so we actually immediately, because he was symptomatic from all that disease, he actually was immediately started on combination chemotherapy with capecitabine plus Temodar, had an absolutely beautiful response, thankfully. And then we gave him a treatment break. So to the question before on, you know, can you stop these therapies? It's easier to stop chemotherapies because Temodar in particular is what we call an alkylating agent, and it can damage the bone marrow. And so this patient was, you know, stopped on his therapy and has actually done really well off all treatment. So very different in terms of the treatment approaches there, but actually very, you know, again, same type of cancer, but yet very different because of the biology and the pattern of disease. Okay, so in moving on in sort of understanding that not every patient is the same and that we really do want to make sure that you have a neuroendocrine expert or team taking care of you to help define the right disease, because again, unfortunately, the evidence doesn't show us exactly what to do, like in breast cancer, for example. We really want to get a team that can help use these different approaches, because very often we do all of them, as we talked about on the panel, but the order comes into play, and we'll talk about that in the clinical trials that are going on now. And these are all the different therapies that you heard about this morning that we want to be able to use at any one time. And that's another thing that's really important in making sure that you don't over-treat, because sometimes some of our patients actually do have treatment options left over, but we'll find that their platelets are too low, and so we can't treat them. And so we have to be very careful in trying to think about, like, do we need to stay on this therapy or not? So where are we in terms of the recent trials? So I thought I would start off with somatostatin analogs, and I'll call it a cute little study we did that was just recently published in the Journal of Clinical Oncology, Clinical Practice. Because octreotide and Lanreotide, I think it's important to know, are biochemically the active site, meaning the active part of the drug, is exactly the same. So people say, no, you have to use Lanreotide because that data shows it's better than the Octreotide. The reality is they're actually biochemically the same active drug. The difference is one is a deep sub-Q, the other one is intramuscular. But the studies had very different patient populations. So you can't say, well, Lanreotide had a better outcome than Octreotide because they were two different studies at two different times. But nevertheless, we asked the question, which one do patients like? Again, we're going back to that quality of life thing, because one is sub-Q and one is intramuscular. Could we randomize our patients in a blinded fashion and give them 3 doses of Octreotide, then 3 doses of Lanreotide, and every month ask them, how do you feel? What was your pain? And on a scale of 1 to 10, which one did you like or not like? And in the end, which was not so surprisingly, at least in our case, there was no difference in pain scores and over time the pain itself. But there are differences in cost, which is important depending on your own insurance company. So it's not a one-size-fits-all on which one is going to be cheaper. But it's just something to remember. So again, this was a quality of life study that essentially showed that in terms of patients' quality, there was no difference there. So what else is new? So just recently, about to be on the press in Journal of Clinical Oncology, Dr. Kuntz for pancreatic neuroendocrine cancers. Many of you that have this type of disease have probably been on it or know about it. Tested the utility of whether combination oral chemotherapy is better than single agent, and the combination won out. And so combination capecitabine plus Temodar was better at controlling the cancer, almost 2 years, thankfully, when you had combination versus 14 months for Temodar alone. And so this is a combination therapy that we often use for patients with pancreatic neuroendocrine tumors. Of note, another study just recently came out to say if you're on Temodar for longer than a year, it can significantly and irreversibly hurt your bone marrow. So really, again, this is one, because we have so many other important therapies we want to continue to give at some point, have to be very careful to not stay on that drug too long. So that's important drug combination therapy that came out just, or is coming out now. What else do we have? So the next study is lung nets. So for those patients that may have lung nets, this is a really uncommon cancer. I'm going to talk about one trial that we have right now that looks at PRRT versus everolimus that we have. But for lung nets, the most recent trial that came out was looking at Lanreotide versus best supportive care. So does somatostatin analogs help in lung nets? Because almost all the data were in the so-called GI neuroendocrine cancers. The study, unfortunately, was closed to accrual because it was very hard to do a randomized study when one of the arms is just placebo. But with the small numbers, it was a positive study. And so it is on the guidelines that somatostatin analogs can and should be used in patients that have somatostatin receptor positive lung neuroendocrine cancers. So this study also came out in the last 6 to 12 months, and I thought was really important. It's a very important drug for our patients that have a germline mutation called VHL. So pretty unlikely that anyone would have this, because it's probably less than 5% of our patient population, probably less than 2%. But a critically important drug because it goes right after the money. So for patients with this germline mutation, they can often get kidney cancer as well as pancreatic neuroendocrine cancer and other problems of the spine. And when they actually randomize patients to get this drug, those that had pancreatic neuroendocrine tumors had an 83% response rate. So it sort of melted it away. So it's FDA approved for patients with that germline mutation. So again, unlikely, but this is if any family history suggestive of neuroendocrine in the family or kidney cancer in the family, it's something that we definitely want to get tested so that our patients can get this drug if needed. So what about immunotherapy and neuroendocrine? That was a patient question, which is an important one. And as you heard, immunotherapy works by turning on our own T cells, which is one of our white cells, our infection-fighting cells. And by turning on our own immune system, it can actually attack the cancer. And so several immunotherapies are now FDA approved for many different cancers. The problem with our cancer is several studies have failed to show that it can turn on our immune system to attack our cancer. So it hasn't really been so helpful. However, this year, just a couple months ago actually, this study that I talked about on the panel did show that when you used a certain type of immunotherapy called atezolizumab, with a drug called Avastin or bevacizumab, there was a positive response. So they had 40 patients, 20 of the patients had neuroendocrine in the pancreas, 20 of them had neuroendocrine that didn't start in the pancreas, either GI or somewhere else, like small bowel, large bowel, or lung. And they essentially showed that they had a 20% response rate if it was in the pancreas and a 15% if it wasn't. So not huge numbers, in terms of the tumor shrinking. I will say, for the purposes of understanding clinical trials, when we say a 15% response rate, in order for a clinical trial to tell you that you responded, the cancer had to have shrunk by 30%. So when you see these numbers, that means that the cancer shrunk by 30% or more. If you had your cancer shrink by 15 to 20%, you're considered stable disease. So these numbers, although small, doesn't mean that nobody got benefit. It means that the small numbers of patients actually had that major tumor shrinkage. So it's exciting, but again, the problem is because it wasn't randomized, we don't know what if it was just the Avastin, or what if it was just the immunotherapy, or what if it was the combination of both. So exciting that there could be a possible further evaluation of this, but the data is still too small, unfortunately, to make it to the guidelines to be considered. So more to come. I think most of us, when you look into data of like what types of cancers respond, for example, lung cancer, those cells tend to be a little bit more aggressive, and they tend to have a lot of what we call mutations or genetic damage. Our tumors, probably in part because they're so slow growing, have very small numbers of mutations. So the theory out there is because, the good news is our cancers are slow growing. The not so good news is that they don't have a lot of mutations and they don't look foreign to our immune system, which is probably why they don't work as well. So the great Dr. Liu is going to talk about this, but I did, before talking about clinical trials in peptide receptor radiotherapy or PRRT, I just thought I would give a quick tutorial, and then I'm sure you'll hear about this again with Dr. Liu's talk. So peptide receptor radiotherapy has actually been around in Europe for several decades. It took us a couple decades to catch up to them. But as you know, we talked a lot about the PET galliums already. So what is important about sort of this PET imaging and PET treatment? So we talked about this morning how getting a PET gallium scan helps us look for the somatostatin receptors, and when they light up like a Christmas tree. And very importantly, that helps us for what we call prognosis. So tumors that are very positive, those patients tend to live longer, thankfully. And then equally, if not more importantly, it allows us the opportunity to use peptide receptor therapy, which will actually bind to that receptor, and then the radiation itself will get internalized and then zap the cancer and kill it, which is great. So but there's different, so the traditional PRRT that we know is lutetium-177. That's the radio-labeled isotope that's on the therapy that's FDA approved for neuroendocrine cancers. But there are several others, and I put them here, that could be used as a radio-labeled tag, if you will. And so I just have a little schema here of the so-called traditional radiation therapy that we always know about as sort of one beam that goes to one place. If there's like a bone metastasis or something that's painful, we will use that. But on the right is just a little schematic that I took from Dr. Jason Lewis, who does a lot of this work with me at MSK, and shows how when you go through that PET scan, we're giving you that IV, and then the PET scan camera will take pictures, and then all over the body, wherever it's lighting up, is where the radiation and therefore where the disease is located. So again, Europe has actually proven that peptide receptor radiotherapy is very important, both for diagnosis for those PET scans that we do, as well as for treatment. And that eventually led to its FDA approval here as well. This is a study that I did with one of my colleagues at MSK, Dr. Weber, on a different type of PRRT called JR11. And you could see not only is it helpful for showing the disease, but the different images there showed that this was a complete response in one of our patients that got this therapy. This is a drug that we were very excited about, but is no longer going to be tested. However, the new kid on the block that I'm sure many of you heard about, and that Dr. Liu will probably talk about a little bit as well, is the so-called alpha. So a different type of radiation that's linked to the somatostatin receptor, and Why is it so cool, and why has there been so much excitement? Well, most of the data comes from prostate cancer. So in prostate cancer, when you link that little bit of radio-labeled isotope to PSMA, which is a protein for prostate cancer, they showed beautiful responses. And importantly, what I will show is that this was after lutetium-177. So this picture here shows you a complete response. If you can see, so that picture on the left is a PET scan of a patient with prostate cancer. And look at all that black, that's all disease. They had already received lutetium-177 PSMA, so that would be like the equivalent of our Lutathera, lutetium-177 somatostatin. And then 2015, and then 2016, they get the alpha PSMA, and the patient has a complete response, which is, again, very exciting, and something that all of us are potentially hoping and expecting to have. So what are the clinical trials now with all of that in mind for PRRT alpha? There is one trial in Texas, as some of you know, which is targeting therapy for patients in naive neuroendocrine cancers. So very importantly, though, if you talk to nuclear medicine folks that have been using these different types of therapies, et cetera, they're a little bit reticent to do it in neuroendocrine cancers. That's not to say we don't want to do clinical trials. I mean, this could be a very exciting drug for us. The problem is there could be potentially more kidney toxicities and issues. And we don't know for sure. Again, in prostate cancer, first you got the lutetium, and then after that the alpha still worked. But whether giving alpha first before the lutetium is better or not is still unknown. So these are one of the issues when we talk about clinical trials and we talk about how courageous patients are to go on these studies, because we just don't know. So this is the only study right now that's available that I'm aware of for alpha in the United States. There is another imaging study that's available in Iowa, but it's only for imaging. It's not for treatment. So that's the studies available for PRRT as it relates to alpha. What about other issues? So at MSK, we are in the process of developing an alpha program. We're very excited about it, but we are actually not ready for prime time at all. So we're starting to develop the program. Again, we've done some with alpha prostate from pharmaceutical industries, but we don't yet have the appropriate trials to do that, but we are very excited, and we're testing a lot of this stuff in the laboratory. So moving on, what are some of the other therapies? So a lot of us, like in the way that we do clinical trials, we take a really good drug like PRRT, which is now we're talking lutetium-177 or Lutathera with Octreotide, and can we add drugs to it to make it better or not? So that's what some of these drug combinations are doing. They're combining it with combination capecitabine plus Temodar with PRRT or alone, or just capecitabine is another one that's going on, or they're comparing it to everolimus, which we'll talk about a little bit. There are some studies in Europe that are actually even looking at PRRT prior to surgery, which could be interesting. But again, none of these are yet something that is prime time or going on. There is one study of PRRT in the adjuvant setting where after surgery they're giving that. That has been very, very slow. Excuse me, that's for capecitabine Temodar. The PRRT after adjuvant therapy, I don't think there is one that's open right now. So there's only chemotherapy for pancreatic neuroendocrine tumors, but in the pre-op setting, they're testing that in Europe. Okay, what about somatostatin analogs? We talked about how Octreotide or Lanreotide are generally first-line therapies, and there are several studies that are also adding that. I think most of us are thinking that this isn't as exciting as we would like when we're adding these different drugs. And again, because we personally want to use these drugs and sort of milk them for as long as possible, instead of combination with somatostatin analogs, it's probably better to do it sequentially, but that some of them are looking at that. I think importantly, in terms of liver-directed trials, lots of patients will ask the questions, which is better, radioembolization or bland embolization or chemoembolizations? We don't know the answer to that, and most studies suggest it's probably a wash, but there is one randomized study that we're almost done finishing, which is looking at bland embolization versus chemoembolization. And so that'll at least give us an indication. There was another arm of what were called drug-eluting beads that had a little chemo on it, and it was way too toxic. So that arm was closed, and nobody should be using drug-eluting chemotherapy beads for patients with neuroendocrine cancers because of the toxicity there. So I think the neuroendocrine trials as it relates to combinations of therapies are important, but many of us feel like the combination may not be as helpful as just sequentially, again, trying to use one drug and then moving on to the next. So we talked about immunotherapy trials, but there still are others that are going on. So even though we haven't been as positive as we would like in neuroendocrines as it relates to immunotherapy, people are trying to manipulate the system, if you will, and try to find ways to turn on the immune system and then give the immunotherapy. So these are a couple of those where they're using combinations of different types of immunotherapies together, plus minus, for example, somatostatin analogs, etc. So unclear if this is really going to do too much, but those studies are still ongoing. I'm not aware of any in the New York State area right now for immunotherapy in well-differentiated neuroendocrine tumors. And then lastly, the sort of other category of what we call small molecules or targeted therapies are important. So there's a drug called cabozantinib, which is an oral therapy. We do have this opened at MSK for any patient with either pancreatic or GEP-NET, a GI neuroendocrine that have progressed on somatostatin analogs and or other therapies. So there's no limit on what therapy you need to enroll in this study. It's an oral drug. It is randomized, so it's 2-to-1 that you'll get the drug. So there is what we call a placebo arm, if you will, on this, but if you're on that and you do have progression of disease, you're able to cross over and get the drug after that. And it's there has been shown to be good activity. So it's a phase 3. So if it's approved, then it would get, if it's a positive study, it would get FDA approved. So the earlier studies did show activity, and it's pretty well tolerated. It's an oral drug once a day. And so in most oncology conferences, it's not a talk if you don't talk about immunotherapy, so we did that one. And then next generation sequencing. So what is next generation sequencing, and is it important or not for our disease? So we talked about just earlier how the more mutations, the higher the chance of a good response in immunotherapy. But unfortunately, we don't have a lot of mutations in our tumors, and equally, those mutations sometimes, depending on the type of cancer, can actually be targeted themselves. So here I have a couple of mutations that are actually targetable, that in different platforms, so for example, Caris or Foundation One, at MSK we use something called Impact. If you have those mutations, we can actually target it with drugs and put you on clinical trials and see if they're helpful. And some of those, like the ones that I have up here, have been shown to be highly effective. The problem is when we did big studies in neuroendocrine cancers, we didn't find a lot of mutations that are targetable. It's probably on the 10% range. However, if you find it, it could potentially mean something. So we did find recently that the so-called poorly differentiated neuroendocrine cancers definitely should get next generation sequencing because they may have mutations that are important to potentially target, or higher grade tumors. So we're often, there was a question earlier on, should I re-biopsy my tumor? There's probably not, outside of a clinical trial, and we do have a clinical trial at MSK doing this, where we're biopsying to really test the utility of doing that. But to date, there really has not been that we're finding more targetable mutations. It's not to say we couldn't try in the context of a clinical trial, but we're not as, unfortunately, lucky, if you will, as other diseases where you can find mutations. Having said that, TSC2, for example, is a trial that we have at MSK that we've been very excited about, and that is something that in our patient population you might see. So that's a mutation that could be very helpful for us. So for pancreatic neuroendocrine tumors, we do encourage patients, particularly if they have higher grade tumors, to look for next generation sequencing to see if we have some of these mutations identified or not. We should not have a conversation again without, or at least a talk without talking about quality of life because of our patients living so long. We really do want to do more studies and thinking about quality of life, and so I wanted to share Another study that we just recently opened is, unfortunately, the oral octreotide is not yet in clinical trial development, or it's in clinical trial development, but the trials are not open. But there is a subcutaneous testing that's going on that we do have at MSK that's offered to test the efficacy and safety of giving octreotide subcutaneously so that you can actually do it yourself at home, which is kind of nice. So instead of having go to the infusion center once a month, some people are already getting a nurse coming to their home that their insurance company is doing, but this is asking, can we do it by sub-Q as opposed to IM, and then having our patients self-inject to see if that can be helpful. And then there are other studies that are focusing on fatigue. You know, a lot of our patients will complain of like grade 1 to 2 fatigue. Kind of a big deal when you're living with that all the time. And so we really want to get a better understanding of like how much fatigue do our patients have, and is that cancer related, treatment related, or both? So then last but certainly not least, what's the best sequencing of all these different treatments that we have that you heard about this morning? Like what should be first, second, and third line? And unfortunately, in other diseases like breast and prostate and lung, we know exactly what needs to be done first, second, third, and fourth line because there are studies that allow us to know because they did it and they said this patient lived longer, therefore you do this as A, B, and C instead of DCBA type of thing. We don't have those studies, but we're trying. And so these are the studies that are going on that actually test the question of if you, for example, gave everolimus first versus PRRT, who does better? So that is, for example, a study that we have for LungNet that's here at the bottom. That's an alliance study. The NETTER-2 is actually, this is a higher grade phase 3 study where they're looking at high dose LAR versus traditional Lutathera. Again, is it better to do like a higher dose first before going to PRRT? This one is everolimus versus streptozotocin, which we don't even use that much anymore. But these studies are going to hopefully help us answer the question of, does it matter? Does it matter what you give first, second, or third, or is it a wash? I think many of us believe that it's probably a wash, but very, very importantly, that we make sure that our patients get them all. So again, these types of studies will help us really answer that question, but we want to make sure that our patients are potentially getting all these therapies, which is most important. So the treatment landscape, thankfully, has changed in the last 20 years, but because we don't have the predictive markers to know, like for any one patient, what is the best therapy and how do you give it, these trials will hopefully help us answer that. And a lot of the stuff you've heard this morning, but I think first that these therapies are all very viable treatment options. And so a lot of what's going on now is not looking at new therapies. I mean, cabozantinib, and there are other small molecules that are out there that still are looking for FDA approval. But as you heard from the talk, most of the clinical trials that are going on right now are saying, which one is better first, or what about the combination of these therapies as opposed to actually new, true, novel therapies, which isn't to say we don't need them, but I think most of the landscape right now is just looking and asking about combinations versus when to give what. And so this is our clinical team at neuroendocrine at MSK, but most especially I just wanted to thank all the patients and the caregivers for participating in the trials that we have. And that is it. I'm happy to answer any questions.